Hi everyone, my name is Hosung, and I'm very glad to be a part of this fascinating workshop. My talk is about the recent techniques used for the motion synthesis and retrospective correction for motion artifact on brain MRI. The reason why we are interested in correction of motion artifact is this is one of the most common artifacts on brain MRI and other body scans too. But I'm going to focus on only the imagings with brain because this is my main field. So how popular this motion artifact is about 20%. You can see from pediatric MRI, 10% from dementia, and even more for the Parkinson's disease and motion movement disorders. And you can see even more within the neonatal and infant brain MRI, and it gets worse when you have ADHD. So these are the very typical ringing artifact in relation to motion artifact. And this pattern of artifact is very different from other types, as you can see here. And there are two different causes for the motion artifact on brain MRI. First one is a free head rotation, which is actually related to the ringing artifact and generally affect the whole brain. Whereas the respiratory motion is more local and affect more like down on the brain, like a temporal lobe and the cerebellum. Why do we care for motion artifact? Because it affects some processes needed for the quantification of brain changes and analysis in terms of statistics later. So the basically on the structural brain MRI, you can actually use tissue segmentation and furthermore measure volume, cortical thickness, curvature, gyrification index, or other types of metrics to understand better about changes of the brain in relation to development, aging, biological hypothesis test, or clinical conditions. And you can see that when you have a motion artifact, the conventional segmentation method can be easily failed and create the corrupted the brain pathology that actually led to changes, nothing to do with the anatomical changes, but more actually make a problematic in urinalysis. So my talk consists of three different parts in overall workflow. This is the quick overview of general workflow. You can see that we need clean data that you can identify by seeing it on your eyes. If there is minimal or no sign of ringing artifact, we can take it as clean data and ground truth. And we need paired data that are from the same subject as clean data, but introduced with the motion corruption. Then finally, we need the another component, usually like using statistical models or machine learning techniques that actually regress the motion patterns and predict the clean data out of the input data of motion simulated MRI data or real motion data in test. The first, we're gonna deal with the some techniques used for the motion synthesis or simulation. Why we need them? Because the regression models based on machine learning, deep learning, they are very powerful, but require a large data set for the training process to avoid any kind of overfitting issue. However, it is very difficult to find or acquire brain MRI data with real motion, real head motion. So the solution is, well, it has been like mathematical models that actually mimic realistic artifact of ringing pattern of artifact due to the motion. So this is one solution, very popular for the recent papers. You can find from like 2017 to 2021, and you see that the you first need to transform the input volume and rotate them using rigid body transformation in different directions. Then that create these images after applying a Fourier transform. You can actually obtain case-based data like that, and you place the original data to be in the middle, and other data actually place in other parts. If you have more data, more transformation, then you can actually proportionally change them. They are the region of interest, and then combine them within the case space. Then at the end, what you're gonna have after inverse transformation, that the data looks like data with the actual realistic motion. Another method that we actually adapted in our paper published in 2021, Neuroimaging, is directly applying some kind of mathematics, disrupting the phase lines on case-based data. So basically, you create some artificial signals on case-based with the lines that goes along with the phase shift. So and we actually change the amount in terms of number of lines and the significant the severity of the magnitude of the lines that actually introduce different severity of artifact in the final data. Once you combine the clean data with this simulated motion, then after like inverse transformation, you're gonna see like how the data looks like with the different amount of like the simulated motion. And very recently, we are still working on to get the better optimization in our method, but we look for some ways like deep learning approaches to augment motion affected images. We have tested two popular approaches like cycle-gang or neural style transfer. Let me introduce them here briefly. So cycle-gang is actually a very interesting algorithm that actually learns some patterns that doesn't need to be paired between two group of images. So for example, like if you use the horse, and you can actually match the pattern and see if there is a major difference between these two images, like stripes on zebra, right? And they don't need to be a same kind, same location of horses. Yeah, you just like put one type of data, like a real photograph. The other one would be like paintings. Then you can the cycle-gang can find the major pattern difference. So in the same way, what we did is basically we take group one as motion affected data, and the other one as motion free data. And the subjects, they don't need to be really paired. Then once the cycle-gang learns the main difference between these two, which is assumed to be motion artifact, then you can actually go back and forth between motion-free data to motion artifact data, or like motion corrupted data to motion-free data. So this is our result so far. You can see like on the left side, from you is the original data with no motion, and the right side of each of the input, they are like generate data with more motion. You can see that all the data in generated images, they look like blurry data. However, at the same time, you can also recognize that the pattern of corruption in relation to motion, it has to be somehow ringing artifact with the anatomy somehow preserved on the image, but this one is not realistic very much. So what we tried next was using neural style transfer method. So here it's more feed-forward method where you use one input data as content, the other one used for style adding. Then output would be preserving the major portion of the data, but in with the different style. So what we did here using that method is basically we input content image as clean data and put unpaired a brain MRI data with somehow degree of motion. Then this is the result after iteration of some number of iterations. The problem here is whenever you do this to get the output, there is somehow a separate training and iteration. So it's more time consuming, but you can see the result looks more promising compared to PsychGAN, generating a more realistic pattern of motion artifact. Then we're going to jump on to the next part, which is the how you correct the motion artifact on brain MRI. So the national for this is, there is interactive prospective method for motion correction. If you have original case-based data along with the spatial image data, or if you have motion estimator, real time, when you actually scan a subject, then you can. However, that's not really the in most of cases. So we assume there is no such data, so that we need to correct after acquisition of the data. So that's what I mean by retrospective. And the training process consists of supervised learning, so that you need ground truth and paired data as clean, corrupted data for the same subject. And output in testing is predict clean data. And I'm going to deal with two different methods. One is using the convolutional network. The other one is GAN, and see which one is better, or what actually they do and how different they are. So this is one method that we actually published very recently using the fit for the convolutional network-based approach for motion correction. So this is very straightforward. You input the data with motion artifact. We use the mathematical model to simulate motion. And then the paired clean data was used for the training of the neural network, so that we calculate the loss as the image similarity between these two. Then in testing, you input realistic, real motion artifact data, or simulate data, see if we can actually predict well the cleaning data. And this is another approach using GAN, actually PsychGAN. So as I explained earlier, this model can simultaneously allow data augmentation and motion correction, meaning that in one cycle, you can input motion-free data and predict the motion-affected data, or you can input motion-affected data to predict the real motion, the motion-free data. So they used that and evaluated using the brain tissue segmentation, and found that it improves in the accuracy of the segmentation once the data was corrected. Even if you can see that in the middle, the result doesn't really perfectly, completely correct the motion because of maybe a different pattern of motion and the excessiveness of motion artifact. And their sample size was relatively small. But PsychGAN did the job that it has to be. Then we actually asked ourselves the question, Is GAN actually useful or even better than the straightforward CNN for the clinical application or biological interpretation? And what we found is that, yeah, this is the original data with motion, and this is just blurring using the Gaussian kernel, and this is using the feed for the CNN, and this is GAN. And you can see after correction with the three different methods, you can see that the results look similar, and these look much better than Gaussian, of course. However, when you look at the actual voxels with zoom, you can see the tissues look more like enhanced in GAN method, which look pretty okay at the first look. But when we measure the some indices from the images, we found that this one is less similar to the ground truth image than the one that corrected by CNN. Whereas, like when we calculate the gray matter white matter tissue contrast, well, the GAN actually unnecessarily enhanced too much of the contrast. And then this is last part of the talk. I'd like to discuss a little bit about like how we can evaluate the performance of motion correction using whatever the method or like your own method. So one way is like, yeah, visually check and you like score for better, good, excellent, something like that. Yeah, and I'd like to show you some ways to quantify the quality of image after correction. So basically what we need is clean data to be compared, of course, and then you use the data before and after correction. And the first measurement that we use very popularly is SSIM, Structural Similarity Index. Basically, like you subtract two images and find how similar these two are. So it goes from like 0 to 100 or like 0 to 1 or like 100%. That's what I mean. And you can see that the after correction compared to the blue, which is the data before correction, that actually showed better similarity with ground truth. But using the more severity of motion, you can see that, yeah, there is like amount you can correct, but there are still always like residuals. Another method to quantitatively evaluate the performance is when you're given real data where a subject is scanned and rescanned with the intended motion and without motion. This is one of the ideal case to evaluate how well the motion correction method works for real motion corrupted data. However, this way is limited by small sample size because getting this scan rescanning is very unrealistic in actual research setting. However, we are luckily found that there are like a small size of data, like 13 subjects in Alzheimer's disease neuroimaging initiative data set who are basically initially scanned with motion so that they actually have to go over another session of scanning so that there were like two scans without motion. And we found that using the CNN-based correction, indeed improved the image quality in terms of the image similarity between the reference image without motion and the corrected image compared to the before correction. And also the signal to noise gets improved. There are a couple of ways to indirectly evaluate the performance of motion correction. One is we go over some pipelines like free surfer that actually extract the brain tissues and the borders of the gray matter, white matter, and CSF tissues that create cortical surface, which is called the surface reconstruction. And then once you do before after the motion correction, we can run over some visual quality control that identify locations with the errors in surface reconstruction. And we found that the after correction, there are many places showing defect in the data with motion without correction. You can see that the about 20% also who used to be considered complete fail or questionable in surface reconstruction converted back to pass to be like good quality for the analysis. Finally, we like to understand, even if you observe so far, the motion correction method can actually improve the quality of images. However, we don't know yet if they actually improve the data in relation to anatomical and biological interpretation. So whether the when artifacts were corrected, the anatomical and biological information, are they also washed away or like very well preserved? And what we actually found in statistical analysis on the cortical thickness measurement to compare between the changes between the health controls and Parkinson's disease, we expected to have changes in the association cortex and the frontal lobe and temporal lobe cortical thinning. And we found that actually in the data before motion correction. However, after correction, we found the more effect size of changes and include the more extended area with cortical thinning, which actually made a lot more sense to us just because of like a different data quality. And we proved that the correction method doesn't really wash away important biological information. So finally, in this talk, we dealt with the some ways for the data augmentation of motion artifact or like simulation of motion using a mathematical model. Also, we looked for some ways that actually to augment motion artifact data using machine learning method, and they are very promising. We still need to run some qualitative and quantitative evaluation, see which models actually show more realistic data augmentation. And in each situation, one method would be more like useful than the other one. And then we looked for the different ways of motion correction method and compared between the feed-forward convolutional neural network versus GAN, and found that GAN is very nice for the data augmentation synthesis of motion artifact. However, some overestimation or like because of the behavior of a discriminator in GAN that actually find the realistic data changes in machine learning may not really capture the biological information, then sometimes it over-enhance the data. So may not be very good solution for some clinical applications. Finally, I would like to indicate my special thanks to the lab members involved in this research, and also the faculty collaborators at the USC Zuni, also the funding sources used for like the performance of this body of research. Thank you so much for your attention. I will be very happy if you have any question and I'd like to answer you.